I'm going to talk about some work that we've been thinking about in the context of what we call biobanks. So I'll tell you a little bit about this work. So for a lot of time, we've been interested in these data sets called biobanks as a way of learning about human genetics and how it contributes to disease and phenotypes. So essentially, biobanks are these repositories which are bringing together genetic data combined with very diverse, rich phenotypic data. And what's happened over the last 5 or 10 years is really there's been this explosion in terms of the diversity of the kind of data that's being collected here, but also the scale. So we have biobanks here at UCLA, but also at other academic institutions across the US, Europe, Asia, and so forth. And we now have biobanks where the scale of the data, where the number of individuals that's collected, is typically of the order of hundreds of thousands, some approaching even 1 million. So this is very interesting, and this brings with it some really interesting challenges for us as quantitative, statistical, computational people. And one of the things that I'm personally interested in is how can we learn about complex traits, the link between genetics and complex traits, now that we have these data sets with millions of genomes linked to rich phenotypic data. So this is a multidimensional problem, and there are lots of ways of thinking about it. One angle is the statistical angle. The data that we get here is messy, biased in lots of different ways. So how do we build statistical models that really account for all of these sources of bias while extracting the signal that we like? The other important challenge is the data access itself is not very trivial. There are lots of issues for how we access the data, what type of data we can access because of privacy concerns. So how do we work with these types of restrictions? And finally, the scale of the data, that engenders issues of running these methods at the scale that we care about. And so that brings with it computational issues. So really, there are all of these different issues, and often they're in conflict with each other. There are trade-offs. So how do we really go about thinking about these different issues is a grand challenge for us. So not all of you might be familiar with biobank data, so let's just orient ourselves. So just to make things simple, the data we are going to be looking at is data that looks like this. So we have individuals here. Each row is an individual. And each column, in terms of their genetics, represents the genetic variation of an individual at a particular position. So this is typically a single nucleotide polymorphism. So each individual is carrying one possible variant at that position. And because we are all diploid genomes, so we represent each individual's SNP data as an entry which takes 0, 1, or 2. So encoding whether you carry 0, 1, or 2 copies of that variant. And now you have paired with it a phenotype. Again, for simplicity, we can think of phenotypes as scalar valued, but of course, there is imaging data, there's time series data, but we'll start with a very simple representation here. So this is a very typical biobank data set. And now let's think about what are the kinds of questions, the key inference problems that we'd like to solve here. So there are 2 types of inference problems that we'll focus on. The first one is simply looking at the genetic data, the genotype data, and trying to understand what are the relationships between the rows of this matrix or the individuals in this data set. And the reason why we care about this is because these types of relationships are informative of recent relatedness, for example, families, or more distant relatedness, for example, populations or ancestries these individuals might belong to. So broadly, this type of problem is what is called population structure inference. And I won't say too much about it in this talk, but a key question here is how do you infer these structures, population structure, which ancestry each person belongs to, from this type of genetic data? And how do you do it at scale when you have a data set with, say, millions of individuals and millions of SNPs? So we have worked on these types of problems in the past. I've talked about it at past CGSIs. And the key idea here is this notion that we can think of this matrix of genotypes as having a latent low-dimensional representation. So we can write down this genotype matrix as a product of these lower dimensional factors. And we like to be able to learn or decompose this genotype matrix into these low-dimensional factors. And now, depending on the types of assumptions that we are willing to impose on this low-dimensional factor, you end up with different types of models. For example, if you impose orthogonality, you end up with something called PCA, principal components analysis. If you impose other types of assumptions, for example, non-negativity, living in a simplex, etc., you end up with something called admixture or topic models. So for example, there was a lecture by Sushmita Roy on non-negative matrix factorization, which is very closely related. So the question is, how do you infer these models? And that's challenging. And the key idea is we're going to view these different types of models as instantiations of what we call latent variable models, where these quantities here, which we don't observe, are unobserved. And we use insights from randomized linear algebra to speed up the inference of these types of quantities. So I won't say too much about population structure beyond just this, and a lot of this has been published. Now I'll move on to the second inference problem, and that's what I'm going to spend time on. And here we are interested in making connections between the genotype and the phenotype. Broadly, we think of this as the problem of inferring what we call genetic architecture of these traits of phenotypes. And we often care about what we call complex traits or phenotypes. These are traits that are determined by many possible regions in the genome interacting with environmental factors. So what do we mean by genetic architecture? Broadly, there is some function that's mapping a person's genotype and environment to the phenotype. And we like to be able to see what this function looks like. Again, we are pretty abstract, so we can break this down into lots of component problems. So one problem might be to identify what are the specific genetic variants that are associated with the phenotype. So typically, this is the problem that is being answered by a genome-wide association study, or a GWAS. There are other aspects that we might care about. For example, how much of the phenotype can be explained or predicted by genetic variation. So this is a concept called heritability. We might be interested in how genetic effects are modulated by context. Context could be environment, so that leads to gene-environment interactions. Or it could be other genes, so that leads to things like dominance or gene-gene interactions. Or we might be interested in how these genetic effects are shared across different phenotypes, and that leads to something called genetic correlation. So at the end of the day, when we are thinking about genetic architecture, we are talking about specific parameters in some statistical model, and we like to make inferences about these parameters. So just to start simple, let's look at this concept called heritability and see how we might go about estimating it. So to start out, we are going to assume a very simple model that relates genetics and trait, and this is going to be a linear additive model. Why do we start with a linear additive model? Do we think that that is the true nature of genetic architecture? Probably not. However, it has a lot of useful properties. It's a fairly simple model to reason about. And even this model is going to illustrate some of the challenges when we move to biobanks. So it's a good model to start with, even though it may not completely describe biological reality. So how do we go about estimating this concept of heritability? So we'll write down a model. It's a linear model that links phenotype to genotype, where every SNP in this genotype has some effect that acts additively independent of all the other SNPs. And everything else that we cannot model, we simply term it environment. So this is the simplest model. So phenotype is a linear combination of genetics with some effect sizes plus noise. So this is statistics 101. Estimating a linear model is the simplest problem you could think of. That seems simple. But the challenge is we are still in what we call a high-dimensional setting. So even with millions of samples, the number of SNPs that we are dealing with is often an order of magnitude bigger. So to be able to estimate these effect sizes is not possible unless we impose additional assumptions. So we're going to impose assumptions on the effect sizes of these SNPs. So one common way of going about this is we might say every SNP has an effect size that comes from some distribution. The details don't matter, but we'll say that the distribution has a mean of 0 and some variance that is given by this variance parameter. So we call this the genetic variance component. And then we have the environment, which is coming from another distribution with, again, mean of 0 and a variance that we call the environmental variance component. So now what we have done is essentially we have a model where we are integrating out the effect sizes of all of these SNPs. And we have these hyperparameters that quantify the genetic and the environmental variance component. And these two parameters or hyperparameters together define what we call heritability. So heritability has this definition here. So it's a fraction. It's a number that lies between 0 and 1. And the way you can interpret this is if a trait has heritability close to 0, it's essentially telling us that it has little predictive signal coming from genetics. Heritability close to 1 means it is highly predictive from genetics. And of course, this is not specific to genetics. You might be working with any other type of data, microbiome data, imaging data, and you can still ask the question, how predictive are your features about your response? So heritability is a kind of genetic signal that is relevant to your trait of interest. And so what we're really trying to get at is what is the signal-to-noise ratio associated with genetics when we're thinking of a trait. Fairly simple model, but it turns out even this simple model can be made more biologically plausible. So in this model, we are assuming that every SNP, no matter where it lies in the genome, has the same prior distribution on its effect size. And of course, there's plenty of evidence that suggests that that's a rather simplistic model. So you could easily modify this model and say, I'm going to put a different prior distribution on the SNP effects depending on where in the genome the SNP falls. For example, SNPs that fall within protein coding genes might come from one prior distribution, and those that do not fall come from a different distribution. So now you have moved to a model where you have these additional parameters depending on which part of the genome the SNP lies within. So these types of models that we have just described, they go by the broad name of a variance components model or a linear mixed model. And they've had a lot of influence in the way we think of human genetics and its influence on complex traits. So here is an example of a famous paper from several years back, which was essentially estimating these variance components. And what you can do with this is you can ask, are there regions of the genome where there is much more genetic signal than you'd expect? So it's a way of quantifying enrichment of genetic signal in particular regions of the genome. So for example, you might find that, say, in coding regions or evolutionarily conserved regions, there is an enrichment of this heritability. All right, so this is the model, and we'd like to estimate the parameters of this model. So that's the first inference problem we're going to talk about. So here is the setup. You have your phenotype. We take our SNPs, we divide them up according to some prior biological knowledge into components. So we have k such components. Each SNP that falls in a component has an effect size that is drawn from this distribution. So now you have parameters, a variance component parameter for every such component, and then you have the variance component parameter for the environment. And our goal is to estimate all of these. So that's the simplest estimation problem that we'd like to solve. Again, this is classical statistics. You have a statistical model. You have parameters. We know how to solve it. You write down the likelihood, maximize, right? So straightforward. But now this is where the challenge appears in the context of biobank data. So if you were to try to evaluate and maximize or attempt to maximize this likelihood, turns out it's computationally expensive. So it scales as cubic in the sample size that you have. So n is the sample size, so scales as n cubed. So imagine you're applying this to a data set with 500,000 individuals, 1 million individuals. This kind of an approach doesn't work. There's been a lot of very clever algorithmic work in trying to bring down this exponent, but nevertheless, all of these methods are extremely challenging when you try to apply them to large data sets. So what do we do? So one way to think about it is let's look at an alternative way of estimating these parameters. Again, stats 101, there are two broad ways of thinking of estimation. One is maximum likelihood estimation. The second is method of moments. So let's look at what method of moments has to offer us. So method of moments has a very simple principle. We're going to write down the moment equations of this model. So we'll write down what the mean, the variance, any other moment that we choose to think about. That's going to depend on parameters. And now let's choose the value of the parameters so that the population moments, the theoretical moments, match what we see in the data. Turns out this has been proposed in genetics. It goes to a classical paper by Haseman and Elston. So it's called HE regression, but fundamentally it's a method of moments approach. So let's see if we can apply method of moments in this setting. Turns out the way we have defined this problem, there is no information in the first moment. So the first moment is always 0, so let's move on to the second moment. So you can write down the covariance of y, so that's the second moment. And it has this relationship to our variance components. So those are the parameters. And they are all weighted by these matrices. So this matrix here is often what we call a genetic relationship matrix. So what it tells us is how similar are every pair of individuals at those components, right, in terms of their genotype. So now we have this relationship that defines the theoretical moment. We can estimate the covariance matrix from data, that's the sample moment. Now let's find parameter values that make these two as close to each other as possible. Now what's nice about the method of moments in this framework is there's a closed form solution. So you can actually write down what the expected answer should be. It's solving a linear system. So nothing is easier than solving a linear system. So essentially, we can write out the method of moments solution. But now the question we should ask ourselves is, have we actually got a scalable solution? The answer is not quite, because we still have to compute these GRMs, these genetic relationship matrices. So it's a matrix of n by n individuals. So just filling up this matrix is quadratic in your sample size. And if you ask how much time it takes in building these GRMs, this is what it looks like. So it's quadratic in the number of samples, and m is the number of SNPs. So still not quite good enough for biobank-scale applications. So this is the point at which we were thinking if there is something that could be done that could go beyond the limitations of these types of approaches. And now if you look at it and if you stare at these equations hard enough, you'll see that we are doing a lot of wasteful work. So essentially what we are doing is we are computing these large matrices, these GRMs, n by n matrices, and then we are essentially reducing all of that information into the coefficients of this linear system. Now what are these coefficients? It turns out the coefficients are traces of this GRM or products of this GRM. So you're essentially constructing these very large matrices and then collapsing them into a small number of quantities, and that seems wasteful. So a question is, can we avoid this whole process and directly get at the coefficients, these traces? It turns out we can, and this is what we get from insights in randomized linear algebra. So there's a lot of work in essentially how do you compute or approximate these trace quantities by randomization. So the key idea is you'd never ever actually form the GRM. You simply multiply this genotype matrix with a certain number of random vectors, say B of these random vectors. And as long as these random vectors have some properties, simple ones like mean zero, identity covariance, you form these products and they allow you to approximate these traces. So we just heard a talk from David talking about sketches. So this is exactly a sketch. So what we're using is a sketch or a random projection of the genotype matrix. And what this entails is multiplying this genotype matrix, n individuals, m SNPs, both n and m large, with b random vectors. Now, of course, the efficiency of this method depends on the value of b. So the smaller the value of b, the more computationally efficient it is. The question is, what's the trade-off? How much do you lose in terms of statistical efficiency? How much do your standard errors blow up? And what is interesting and surprising and not quite obvious is this method works for fairly small values of b. So you can choose a value of b as small as 10, so 10 random vectors. So you've gone from a matrix of dimension million by million to, say, million by 10. And that preserves a lot of the information that is relevant for these types of questions. And what's the cost? So now you have to take this m by n matrix, multiply it b times, and that's your sketch. You have this interesting log factor here. And the reason why you have a log factor is the genotype matrix has some special properties. It's discrete, so you can do this multiplication even quicker than other unstructured matrices. So this method, which combines HE regression with randomized trace estimation, we call it randomized HE regression, RHE for short. There's another nice property that's extremely useful, and this method is what we call a streaming method. So you just have to read the genotypes once in small blocks, operate on them, and you do this maybe in a couple of passes, and that's all you need. And so it's a very memory efficient algorithm, which is also important for these large data sets. So we don't care too much about results, but anyways, I'll present a few sample results. So the first question is, how accurate is this? So the gold standard, of course, is maximum likelihood. So we can ask how much is the extra kind of loss of efficiency compared to maximum likelihood. We can only do this on relatively small data sets, so tens of thousands of individuals where MLE can be computed exactly. And so what you see here for different types of genetic architectures, we have GCTA, which is this maximum likelihood estimation approach, and RHE. And you'll see that they are quite comparable. Roughly, there's a 10% increase in the standard errors. We can also ask questions like, how much extra standard error do you have due to the randomization? And there you see, it depends on how many random vectors you use. So with 10, you have about a 30% increase in your standard errors. With 100, it's about 10%. So clearly, with even a small number of random vectors, the contribution to overall standard errors is quite low. And of course, the method is highly scalable. So here, you're looking at what happens as you increase the sample size. These methods here are maximum likelihood methods with lots of clever algorithmic techniques. And this is RHE, and you can see it runs very easily to 300,000 individuals. This was UK Biobank. And we talked about the fact that you can make the model more biologically interesting by allowing it for to partition the genome into these annotations. And again, those kinds of things can be done way easier with this framework than with these MLE approaches. So for example, you could divide up your genome into tens or hundreds of annotations and still fit these variance components across them with extremely efficient runtimes. Now you can apply this to UK Biobank, and you can estimate things like heritability, enrichments, the kinds of things we talked about. Again, not spending too much time on the results. This is all published. You'll see things that have been reported in other studies, like additional heritability from including lower frequency variants that were not included, enrichment of heritabilities in well-known annotations, conserved regions of the genome, coding regions, but also trait-specific enrichments in things like enhancers and so on. OK, so let me pause here and see if there are questions before I move to the next part. Yes?
I feel like I should know this, but how does LD score regression fit in?
Yeah.
I've noticed it's not on the plot, it's very different, but it's calculating related similar things.
Yep. Yeah, so LD score regression, so yeah, I had some figures which I removed. So LD score regression is a class of methods, we'll return to this in a bit, which is using summary statistics. And the way it gains its efficiency is by making approximations about the extent of LD in the genome. So it assumes that LD is relatively local in the genome. And it actually turns out that that leads to a certain amount of bias in these estimates. There's both bias and increase in standard errors. And we'll talk about this further down when here this is a method that's using individual level data. And I'll talk about a method that works on summary level data, which is an apples to apples comparison with LD score regression. Yeah. Yeah, so it's a really interesting question, and we think it has to do with the fact that these are relatively homogeneous populations of kind of distantly related individuals. And so roughly what's happening is you have this GRM, and if you imagine that people are kind of distantly related to each other, the entries in this GRM are quite small. And what that means is you can efficiently estimate the gross properties of this GRM with a few random vectors. Now, the cases where you can imagine this kind of an approach fails is, imagine you have like twins in your data. So you'll have these large entries in your GRM and mixed with smaller entries. And that is the place where small numbers of random vectors are likely to be insufficient. So really understanding the properties of this is not something we have a good idea of, but this is the intuition that's explaining why this method is working. OK, so everything so far was about linear additive models. So one of the things about having these biobanks is really the fact that you not only have a single phenotype that is measured, you have all of these environmental exposures that are extremely interesting. And so the question is, can this allow us to move beyond thinking about simple linear additive models? So can we look at nonlinear effects? And can we estimate these in large-scale data? And kind of the key thing that's interesting about what we've been working on the last few years is this approach of setting up the method of moments, randomizing it and solving it, seems to give really good results across all of these different problems. So I'll tell you a little bit about some of these different models. So the first nonlinear model you can think of, which departs from additivity, is dominance. So in dominance, you have some genetic variant. So you have the 3 genotypes here. You have the phenotype. And if you have some departure from additivity, we can think of it as a type of dominance. We call it dominance deviation. So the question is there evidence for such type of effects in terms of explaining the phenotypic variation in complex traits? So you can set it up as a model like this, where you now have your additive variance component, and now you have a dominance variance component. So you have, again, their effects that are drawn from these different distributions. And so now we'd like to estimate both the additive and the dominance variance component. So we can use essentially the same type of ideas and apply it to traits in the UK Biobank. So here we looked at about 50 traits. And what we find here is there is evidence for substantial additive heritability across these 50 traits. So on average, 20 to 30% additive heritability. On the other hand, in terms of dominance, there is little evidence for dominance. So if you do your hypothesis tests, almost all of these traits have no evidence for dominance. If there exists dominance, it's less than 1% in terms of its contribution to phenotypic variation. So dominance effects are likely to be small. So now we can move on to other types of nonlinearity. So the fact that we not only have phenotypes, but also other environmental exposures allows us to ask questions about interactions between environment and phenotype. So this leads to something called gene-environment interactions. Again, the cartoon picture here is you have some phenotype like BMI, and you're asking what's the effect of some variant on BMI. And if the effect is modulated by, say, smoking, then this would be an example of G by E. So we can take our basic model. So we have the additive component here. And now we can have another component, which is measuring gene-environment interactions. So what we have done here is essentially forming the pairwise interaction terms between an environmental variable and all of the SNPs in the genome. And we're asking, in aggregate, do these pairwise interactions between environment and genome-wide SNPs, do they explain phenotypic variation? So that is encoded in this G by E variance component term. And now, again, using the same basic idea, we can estimate the additive and this G by E variance component jointly on these large data sets. Again, the story is largely the same. There are other methods that have been proposed. And for technical reasons I can talk about offline, many of these methods have inflated false positive rates, even when there is no G by E, whereas our method, which we call Genie, has calibrated false positive rates while also being scalable. We then applied it to, again, about 50 traits in the UK Biobank, and we can estimate how much G by E heritability exists for these traits and particular pairs of environments. So we looked at things like sex, smoking, statin usage, and we can ask for each of these how much is explained by those specific environmental variables. In general, G by E contributions are lower than additive, but they can still be quite substantial. So on average, depending on what specific pairs we look at, it's 7 to 15% of the additive heritability. We can also do things like not just look at genome-wide G by E, but look at G by E in specific regions of the genome. So we can partition the G by E to see if there is enrichment. So here's an example analysis where we looked at tissues, so genes that are specifically expressed in tissues in the GTEx project, and we asked, is there enrichment of G by E signal in those tissues? And we can compare it to where the additive signal is enriched. So here we are looking at additive signal for BMI compared to G by smoking effects for BMI. And here there seems to be some concordance between enrichment of G by smoking and additive effects. On the other hand, other traits, we find that the G by smoking is quite distinct from where the additive signals are enriched. So you can get these kinds of insights into enrichment of G by E by performing these types of analyses. So finally, we can move on to gene-gene interactions. So instead of looking at an environment, you can look at one particular genetic variant, and you can ask, is the effect of this genetic variant modulated by all the other genetic variants that this person carries. So this is a particular type of epistasis that's called marginal epistasis. So that was proposed in prior work from Sheng Zhao's group. And again, it's a variance component model where you can estimate these marginal epistatic terms. The method is calibrated and it's scalable, and we can apply it to these data sets to find significant signals of marginal epistasis. So across these different traits, we find that there are genome-wide significant signals of marginal epistasis. And what is interesting is when you look at the magnitudes of these marginal epistatic terms, they're often larger than what's explained by just the GWAS. So that is interesting because when you're trying to interpret the effect of these variants on a trait, just looking at the GWAS effect, the additive effect, is not quite sufficient or maybe incomplete in terms of the contribution of this variant on the trait. And several of you think a lot about epistasis. Turns out that we can actually replicate some of these signals. So we have ongoing work looking at other data sets to see whether these signals replicate, and to a large extent it appears they do. I'll skip this. So this is joint work with Noah Zeitlin and Val Arbolito's group. And what this is basically showing is for rare variants, there appears to be, again, epistasis with the rest of the common variants in the genome. And some of that might explain things like variable expressivity and variable penetrance of these types of variants. Okay, so we've talked about different nonlinear effects and how we can model these. How do we go beyond the pairwise effects that we have modeled? So is there any way we can model it? Can we look at higher order effects, and is there a way to fit these in the genome? One way to do this is to use this notion of a kernel function. So we'll think of this phenotype Y as coming from this distribution, where the covariance of the phenotypes across individuals is determined by a kernel function. So kernel function is this idea from statistics and machine learning, which is essentially a way of quantifying similarities between your features, your genotypes, in some potentially high-dimensional feature space. For example, if you're assuming that everything is additive, we have an inner product kernel. You're just taking pairs of genotypes and taking their inner products. But you could imagine mapping your genotype to some other space, quadratic, cubic, and so on, and performing inner products there. And so kernel functions allow you to do this in a computationally efficient way. And for example, one very common type of kernel, it would be something like an RBF kernel, where you're measuring the squared Euclidean distance between genotypes and transforming it through this type of a function. So to actually test for these higher order effects, we will now replace our inner product kernel with something like an RBF kernel. And now you can test if the contribution associated with this kernel is 0 or not. So you have a hypothesis test that you can perform here. And how do you actually implement it? You'd have to write down a statistic. And there are many statistics you could write. So the simplest one is something called a score statistic. And this statistic has some distribution that you can test against. The challenge of computing and testing the score statistic is this distribution has a dependence on the eigenvalues of this kernel matrix. So you have to compute all the eigenvalues, and that's, again, cubic in the sample size. So how do we get around this? The idea is, again, we're going to randomize. But the key difference is instead of randomizing over the original data matrix, the genotype matrix, we'll randomize over the Fourier transform of this matrix. So this, again, the reason for this is technical, but the idea is for a lot of these kernels, like the RBF kernel, there is a Fourier transform, and so we'll essentially sample coefficients randomly from this Fourier transform. And so this is what is called random Fourier features, and this allows us to approximate the kernel matrix using these random Fourier features. So as long as the number of these Fourier features is not too big, you can approximate the score statistic. So the statistic can now be computed like this. So D here is the number of Fourier features you need. Typically, for most applications, the number of Fourier features is going to be dependent on the number of SNPs. So you can test small number of SNPs fairly efficiently. And again, the method is calibrated. It scales pretty well. And when you apply it to data and you compare it to methods that are based on a linear kernel, you find additional regions of the genome that appear to have significant associations according to these nonlinear kernels. Okay, so far in everything I've talked about, we talked about this trade-off between statistics and computation and how we can use randomization to get around this. But all of this assumed we needed to have access to a person's individual level genotypes. And in many settings, that's not available. So there are privacy reasons why that's not the case. So the question is, can we now design a method which has both statistical and computationally efficient properties, but does not require access to individual level data? So to do this, let's go back again to the method of moments equation. So this is the linear system that you're trying to solve to estimate heritability. The individual level data filters in through the GRM and the phenotype. And what we can show in some recent work is essentially everything you need to compute the coefficients of this matrix depend on something called the genome-wide LD scores. So this is the relationship to the LD score regression method that was discussed earlier. And the GWAS summary statistics, which are often available for many of these phenotypes. And now we can use our randomization methods to efficiently compute these summary statistics, so these genome-wide LD scores, and the GWAS summary statistics are publicly available. So putting those two together, we now have an estimator of heritability that is entirely based on these summary level data. However, it is mathematically equivalent to everything that we started out with. So this method, it's a version of randomized Huysman-Elston regression that is based on summary statistics, so hence the name. And what we see here is when you compare it to other methods like LD score and SumHer, which is another summary statistic method, this method is much more accurate. In fact, it's about as accurate as the individual level RHE methods. And of course, there are these REML methods, which are a little bit more accurate, but they are computationally expensive. So the key idea is we are now having a method that only needs summary data, is about as accurate as individual level methods while still being scalable. Okay, so just to summarize where we are, so we've talked about some statistical and computational insights that allow us to reason with and work with biobank level data. And one of the key insights is the effectiveness of these random projections. So one of the challenges here is designing random projections with nice properties and understanding when and where they work and are likely to fail. Another key insight is leveraging structure in the data. And I didn't say too much about this, but a lot of this problem, it essentially is multiplying these matrices with random vectors. So you can use these fast matrix vector multiplication ideas to speed up those computations. And in the last part, I've talked about a method that is accurate and scalable while working with summary level data. And this is quite exciting because now these summaries that we compute, you can imagine sharing them across different biobanks. So this can enable things like federated learning. In terms of the biological insights from this, the variance components analyses, what they're showing is that there is substantial additive heritability for complex traits, negligible dominance heritability, and there's emerging evidence for the role of G by E and G by G. Again, additional work will reveal how important these effects are. With this, I'd like to thank all my lab, the people who did the work, several of whom are here in the audience. So Boyang worked on a lot of the G by G and the higher order interactions. Bronson worked on the summary level methods for RHE. And a lot of this work was inspired by conversations and collaborations at the Bertinoro Summer School a few years back and at past CGSIs. Thanks.